Hey there, Dr. Peebler again. We're going to be talking more about mitochondrial heteroplasmy and how it relates to several different disease processes that we all see in our family and friends, and even in ourselves sometimes. So we talked about mitochondria. We talked about that mitochondria have their own DNA system. We talked about that the mitochondrial DNA encodes for proteins responsible for mitochondrial function. And we talked about how mitochondrial heteroplasmy fundamentally works and how it looks, what it looks like, and how there's certain disease thresholds for different diseases of different heteroplasmy states. So we're going to go a little bit deeper into, first of all, the way that the mitochondria are damaged, and then how that affects the bioenergetics and disease processes. And then I'm going to focus on particular diseases that are common within our populations. So we kind of alluded to this, but there's some environmental insult, right? It's an inflammatory insult, it's a reactive oxygen species or redox issue that insults and damages DNA. The DNA, there's a mutation, proteins are not expressed, or the proteins are dysfunctional. Then there is a progressive mitochondrial dysfunction that happens due to a progressive bioenergetic decline in ATP production, which then leads to dysfunction at the organ system level and leads to disease processes. And all of these things are things that are fairly common, heart failure, renal failure, kidney failure, premature aging, inflammatory diseases, cancer, and metabolic dysfunction or metabolic diseases such as obesity and diabetes, all related to these processes. So the first disease that we're going to discuss in greater detail and how it relates to mitochondrial heteroplasmy and mitochondrial dysfunction is type 2 diabetes mellitus. As the paper states, diabetes type 2 is a systemic disease characterized by hyperglycemia and whole organism insulin resistance. The mitochondria lies at the intersection of critical cellular pathways such as energy substrate metabolism, reactive oxygen species generation, and apoptosis, which is also known as programmed cell death. And it is the disequilibrium of these processes in type 2 diabetes mellitus that results in downstream deficits in vital functions, including hepatocyte or fat cell metabolism, cardiac output, skeletal muscle contraction, beta cell insulin production in the pancreas, and neuronal health. Although mitochondria are known to be susceptible to the variety of genetic and environmental insults, the accumulation of mitochondrial DNA mutations, or worsening heteroplasmy, and mitochondrial copy number depletion is helping to explain the prevalence of mitochondrial-related diseases such as diabetes mellitus type 2. The picture on the right is going to talk about how mitochondria are the central problem for diabetes, and how multiple systems are affected by the mitochondrial dysfunction related to diabetes. So diabetes in itself, hyperglycemia can damage mitochondria, and then damage mitochondria can actually lead to worsening glycemic control or hyperglycemia, which then causes the vicious cycle. So I'm just going to pick out a couple of different systems here. And the print is fairly small, so I'm going to try to read this off for us. So basically in the heart, for example, in myocardium, you're going to have a decreased, the red signifies decreases and green is an increase. So you're going to have decreased levels of ATP or energy production. The electron transport chain, ETC, is going to be less likely to be expressed genetically, and there's going to be a less activity of those proteins creating ATP. The mitochondrial density, we're going to talk about mitochondrial density and mitochondrial networks later on. But when you want high energy production, you want to have mitochondrial density and high networks of mitochondria. So when there's lowered mitochondrial density, you're going to have less ATP production. And then PGC1 alpha is something we'll talk about a little bit later. It's one of the main gene transcription factors related to mitochondrial function. We're going to talk about that in detail during the mitochondrial dynamics part of these talks. And then part of the ATP synthase, one of its beta subunits is decreased. In the actual vessels, the skin of the vessels, the vascular endothelium, you're going to have excess oxidative stress. The mitochondrial networks are going to be fragmented. They're going to have a high susceptibility to calcium signaling issues, and induced mitochondrial transition pore opening, and increased caspase 9 activity, which is associated with apoptosis or programmed cell death. And then in addition to that, you're going to have ECT or electron transport chain expression issues, lack of activity, which lowers ATP, et cetera. And you're going to have an issue with induced uncoupling of mitochondria. And then it just goes across the board. So skeletal muscles affected, your pancreatic beta cells, which is where insulin's produced, is affected. The fat cells themselves are affected. The liver is affected. The CNS is affected. The PNS is affected. This is why diabetes is one of the worst diseases you can end up with, because it affects so many different systems. You know, when you have diabetes, they're asking you to go see your optometrist or ophthalmologist, because your eyes are affected, your kidneys can be affected, your vessels are absolutely affected, and your liver can be affected, your brain can be affected, et cetera. So it's a multi-system disease, and the mitochondria play a central role in the progression and the pathogenesis, which is the creation of the disease process itself. So the next thing we're going to talk about is the mechanisms of mitochondrial dysfunction and its impact on age-related macular degeneration. So you're going to see very similar language throughout this, and it's hard to really know where to start in the teaching of these things, because the overarching principle, or the overarching series is called mitochondrial redox. So we're going to talk a great deal about redox and what that means. And central to redox is going to be something called reactive oxygen and reactive nitrogen species. So ROS is short for that. And we're going to have a lot of discussion about how that works, what that is, how there's balance, how there's imbalance, and how the body flexes and deals with that in a healthy and unhealthy ways, which either is going to lead to health or disease. So I'm going to talk. It's going to talk about these things because it's hard to separate the science. Even though we're talking about mitochondrial heteroplasmy here, it's all connected. So it's hard to really separate that while trying to teach a more basic fundamental concept about mitochondrial heteroplasmy without having these words in the system in these papers. So I just don't want you to be confused. So oxidative stress induced damage to the retina, or the back of the eye, the pigment in the back of the eye, the RPE, the retinal pigmentum epithelium, is considered to be the key factor in age-related macular degeneration pathology. Basically, the RPE cells are constantly being exposed to oxidative stress, which leads to the accumulation of damaged cellular proteins, fats, nucleic acids, aka RNA, DNA, which damages organelles like mitochondria. So what this is showing is that the precise cause of RPE degeneration and the onset of and progression of AMD, or age-related macular degeneration, are not fully understood. However, mitochondrial dysfunction, increased reactive oxygen species, and mitochondrial DNA damage, aka mitochondrial heteroplasmy, are associated and observed together and increased protein aggregation and inflammation in AMD. In contrast, functional mitochondria prevent RPE cells damage and suppress inflammation. And the reason that is, and we'll talk about it, is basically, as you can see on the left here, is that the mitochondrial DNA gets damaged. And there's several things that can damage that. I mean, this is a little bit more, gives you a little bit more of a what we call broad differential of things that can damage it. So I'm just going to read these off. So high blood pressure can damage mitochondrial DNA. Eating, there's dietary factors, low antioxidants or low antioxidant systems, just being obese, aka increased inflammation, and generally insulin resistance, being inactive, having cardiovascular disease, smoking, aging, family history of macular degeneration, excessive sun exposure, and having light-colored eyes are all what can damage mitochondrial DNA. And then if you can see there in the central part of that graphic is there's increased ROS, reactive oxygen species, which lead to point mutations, aka worsening mitochondrial heteroplasmy, which then depletes the antioxidant systems and the ability to repair DNA, which then upsets the apple cart. There's no homeostasis or balance, right? There's an excess accumulation of mitochondrial DNA damage, aka worsening percent heteroplasmy in that particular organ system. Then you lose the ability to make energy, you overproduce ROS, and then have progressive bioenergetic decline and mitochondrial dysfunction. And then ultimately the RPE, or the retinal pigmentum epithelium, is damaged to the point where you have retinal degeneration and blindness ultimately. So you can see there's a very stepwise approach about how mitochondria are at play a central role in a lot of the diseases that we are experiencing in our world today. The number one killer of people, at least in the Western world, is heart disease. And a more technical term for that would be atherosclerosis, or the hardening of the arteries. And I think that a lot of people are aware that inflammation is a big driver of that process. In my integrative and functional medicine training, my mentor had something called the 4 horsemen of the apocalypse, or the 4 horsemen of the apocalypse for health. And one of those things is chronic inflammation. And so I think most people know that, having excess inflammation when you don't need it is bad. But the question is, how does that inflammation actually start? Well, it's hard to say, what came first, the chicken or the egg, but it looks like it's the mitochondrial dysfunction that actually leads to excess inflammation. And I'm just going to read this off real fast. So basically, in this review, we will focus on the role of mitochondria and atherosclerosis while keeping inflammation as a link. Mitochondria are the main source of cellular energy. Under stress, mitochondria are also capable of controlling inflammation through the production of reactive oxygen species and release of mitochondrial components, such as mitochondrial DNA, into the cytoplasm or the extracellular matrix, where they act as danger signals, they call them DAMPs, when recognized by the innate immune system. Primary or secondary mitochondrial dysfunctions are associated with the initiation and progression of atherosclerosis by elevating the production of ROS, altering mitochondrial dynamics, energy supply, and promoting inflammation. So, one of the things that I want to do during this whole series is that I don't want to drone on reading scientific papers and having your eyes glaze over. So I've included a ton of pictures that I can hopefully narrate in a way that it makes sense for you all. So we've talked about how there's some stimulus, some thing that's going to damage the mitochondrial DNA, right? Or the mitochondria itself. So that could be, in this case, it's showing oxidized LDL, or some oxidative stress, or even inflammation by itself can do it. And it's going to damage the mitochondria. It's going to then create excess oxidative stress, or ROS. That ROS is going to damage mitochondrial DNA, and it's going to lead to excess inflammation. And the way it does that is through a process called the NLRP3 inflammasome activation, which then activates inflammatory cascade, inflammatory pathways. That's going to be your IL-1 beta here, your TNF alpha, et cetera. And that IL-1 beta, TNF alpha, all those are what they call inflammatory cytokines, are going to kick off, the cascade of inflammation, which then creates damage to the blood vessels and leads to atherosclerosis, or hardening of the arteries. The reactive oxygen species, in addition to kicking off inflammation, is going to actually damage proteins in the process. In this graphic, it talks about how reactive oxygen species damage eNOS, or endothelial nitric oxide synthase, which is an enzyme that creates nitric oxide. And nitric oxide is a vascular antioxidant, but it also dilates blood vessels. And so there's a damage of the endothelial cells, which leads to accelerated atherosclerosis. So I hope that we're starting to connect the dots of how either oxidative stress, redox imbalance, inflammation, hyperglycemia, several other processes can lead to mitochondrial dysfunction through damaging mitochondrial DNA, which then creates excess reactive oxygen species due to redox imbalance, also bioenergetic decline, cellular and organ dysfunction, but also how reactive oxygen species further, in a vicious cycle, damage mitochondrial DNA, create excess inflammation, which then continues the vicious cascade. So in this paper, it's asking, is mitochondrial dysfunction the hidden player in the pathogenesis or the creation of atherosclerosis or heart disease? And it's saying that endothelial dysfunction and lipid infiltration are the primary causes of atherosclerotic plaque progression. I'm going to show you a picture about this later, so don't get too caught up in what that means. In the contest, mitochondrial dysfunction can affect arterial wall cells, in particular macrophages, part of the immune system, smooth muscle cells, part of the skin of the blood vessels, the endothelium, lymphocytes, endothelial cells, causing an increase in reactive oxygen species, leading to oxidative stress, chronic inflammation, and lipid deposition. So this is a paper basically talking about the exact same thing. I'm not going to read this one, but I'm going to read what it says on the right. So there's basically mitochondrial mutations that leads to mitochondrial dysfunction. It adds another dysfunction that happens with this mitochondrial mutations, defective mitophagy. We're going to talk about mitophagy during the mitochondrial dynamics portion of this series. But basically, mitophagy is how mitochondria recycle themselves when they're damaged. That leads to monocyte and or macrophage tolerance suppression, then also leads to chronic inflammation. So what does that look like in real time? Basically, in this slide, it talks about how oxidative stress modifies LDL. The other name for that is oxidized LDL. And when there are normal functioning mitochondria, oxidized LDL is not good. So your immune system is going to try to internalize that, and that's going to put off inflammatory signals. As it says in the normal mitochondrial function, normal mitophagy, you're going to have transient cytokine secretion or inflammatory signals. There's going to be a resolution of inflammation. There's going to be a moderate level of lipid accumulation in the walls of the vessel. And then there's going to be transient immune cell infiltration and then cellular repair. And you can see there on the left that there is intimal thickening, or there's blood vessel thickening. There is a degree of atherosclerosis that's actually happening because oxidized LDL and oxidative stress is a risk factor for the progression of atherosclerosis. However, when you have mitochondrial dysfunction, the same process happens on the right. It's just that there's impaired mitochondrial function. There is defective mitophagy. What you see there, where you start to see the difference, is a constant, instead of transient, you're having a constant cytokine secretion, aka chronic low-level inflammation that's not beneficial to the organism. You have a non-resolving inflammation. It lingers on much longer than it should, which leads to a massive lipid accumulation. And then you have massive immune cell infiltration, slow repair, accelerated atherosclerotic plaque formation, which is highly unstable, which is much more dangerous. This is a very similar picture. So as you can see on the top, you have the normal endothelial cells. However, when there is, mitochondrial dysfunction, you're going to have mitochondrial DNA damage, reactive oxygen species, energy deprivation, and then you're going to have the milieu of cellular and intracellular, extracellular, immune responses to that process, which then leads to hardening of the arteries, thickening, plaque formation, and instability of that plaque, with the potential to rupture and cause a heart attack or stroke. So the next phase of this is going to be talking about heart failure. It's a fairly complicated process, and I think it's going to require its own video, because now we're past 20 minutes. I don't want to take it much longer than this. We've hit diabetes, we've hit age-related macular degeneration, and we've hit atherosclerosis. And we basically tied in how mitochondrial dysfunction due to mitochondrial heteroplasmy, reactive oxygen species, which then snowballs into more reactive oxygen species, inflammation, and basically progressive cellular dysfunction, which leads to organ dysfunction. And in the cases we've talked about before, where diabetes is a multi-system dysfunction, age-related macular degeneration is an eye dysfunction, and obviously the heart disease is going to be a vessel and heart dysfunction. As you can see fairly clearly, mitochondria are playing a central role in all the diseases that are plaguing humanity at this time in our history. I can't imagine any of you don't know someone who has diabetes, someone who has AMD, someone who has had a heart attack or stroke, or has peripheral arterial disease, peripheral vascular disease. These are things that are extremely common. These are things that we see in the hospital repeatedly, several times during one of our shifts here at the hospital. This is something that is definitely should be a concern for the average person, even though these processes are progressive and fairly expected under certain circumstances. There is a lot that we can do to buffer our chances of developing these problems and reversing these problems. So I don't want to be all negative. These are very serious problems, and they can cause a lot of morbidity, a lot of pain and suffering happen from these conditions. And if you can understand that fundamentally, if you can take care of your mitochondria, it's going to take care of your organ systems, it's going to take care of your body as a whole. You'll be a much better position to mitigate the risks and stave off some of these terrible conditions. So I hope that I'm making a strong case of how mitochondrial heteroplasmy is related to these things. And we will talk about how mitochondrial DNA, mitochondrial dysfunction, mitochondrial heteroplasmy play a role in the progression and development of congestive heart failure on the next episode. If you have any comments, questions, please leave them into the chat. If you like this video, you'd like to see more videos like this, please like and subscribe. And until next time.